All right, so continuing with food webs, they can help us visualize relationships among organisms. So this one's nice. It shows leaf fall coming in. It shows algae growing, both feeding stream invertebrates and fish. Then those invertebrates emerging to also feed things like spiders, bats, birds, lizards, and other terrestrial invertebrates. But usually food webs are much more complex like this one. And you need some advanced network analysis to make sense of it. We're not really going to go into the details, but we are going to learn some of the basics of food webs, and they show us these relationships within communities, and they can show us how communities function. The meiofauna are often omitted from food webs, but they can make up to 70% of the total species present. And so it's important that we include these very small organisms as we move forward. Okay, so we're pretty good at the big stuff, and we're pretty good at the really tiny things like bacteria and fungi. We're using our DNA approaches. But the meiofauna are kind of in the middle. So we're going to use stable isotopes for food web analysis. We're going to learn how to use stable isotopes for food web analysis. So here's an ecological pyramid. Shows you we have a lot of primary producers. In general, sunlight coming in, we have herbivores, planktivores, piscivores, and then at the very bottom, we have bacteria and benthic detritivores and organic matter decomposition. This is kind of a typical ecological pyramid with the trophic levels going from low to high at the bottom. Now, this is true for terrestrial ecosystems. We have both more energy, captured, and then more biomass for primary producers, and only about 10% of that energy and biomass moves up into higher trophic levels at each level. Interestingly, though, aquatic systems show an energy pyramid. It's very similar with the 10% going up, but a biomass pyramid that's inverted, which is weird. And so biomass means that there at any given time, there's not a lot of biomass in the primary producers because they're all tiny little single-celled algae, and they have very short lifespans and very high turnover. And so because they're turning over so fast, they can support larger quantities of biomass at higher levels. And so you end up with this inverted biomass pyramid often in aquatic ecosystems. So, but how do you figure out the trophic level, the order, kind of where an organism is? First of all, you could watch it eating. What's it preying on? Second, you could flush its stomach and look to see what it's eaten. The problem with that is that some organisms eat things that they can't actually assimilate, like plastic and wood. And lastly, you could use some kind of chemical analysis to tell what they have assimilated into their bodies. And so the best method is what's called stable isotopes. And so an isotope is an atom that has a different number of neutrons in its nucleus. So for example, we have hydrogen, and you're probably used to hydrogen with just one proton and no neutrons at all. Okay, but if you are hydrogen 2, you're called deuterium, and you have both a proton and a neutron. And if you're hydrogen 3, then you have 2 neutrons and a proton, and you're called tritium. So tritium is a radioisotope. It means it's unstable and it decays into helium-3. But deuterium is stable. It does not decay. And so it's called a stable isotope. Stable isotopes are natural, but they're not abundant. So hydrogen 1 is the lighter, stable form of hydrogen, and 99.9% of hydrogen atoms on Earth are hydrogen 1. But hydrogen 2 is the heavy, stable isotope of hydrogen, deuterium, and it makes up about 0.015% of all hydrogen on Earth. There are the normal kind of light, stable isotope of carbon. 98.9% of all carbon is carbon-12, but carbon-13 is a heavy, stable isotope of carbon, and about 1.1% of the carbon atoms on Earth are the heavy, stable isotope. Oxygen has a few different isotopes. The normal kind of light, abundant isotope is oxygen-16. 99.7% of oxygen on Earth is this form, but there are 2 stable isotopes of oxygen, oxygen-18 and oxygen-17. Oxygen-18 is more common, about 0.2%. And then nitrogen, the normal light isotope of nitrogen is nitrogen-14, but nitrogen-15 makes up about 0.4% of the nitrogen on Earth. So these stable isotopes are natural. There's, nothing wrong with them. They're not harmful, but they're not very abundant. We measure the stable isotope concentration of a tissue using elemental analysis. And basically, we don't have one of these instruments at Evergreen, but it's an isotope ratio mass spectrometer. And it's a combustion method where basically it separates out the different isotopes. It's really good for things like carbon-13, carbon-15, but also oxygen-18 and hydrogen-2. And the measurement is you take the sample, the concentration of the isotope in your sample, and you compare it to some standard, and then you divide by the standard and you multiply by 1,000, and it gives you this delta value. How different is your value from the standard solution using a per mil number? So not a percent, but a per mil, per 1,000. That's what the weird percentage sign looks like. So all of the values are going to be reported in a delta C13 per mil kind of number. So stable isotope, the difference between the isotopes in a tissue occurs due to fractionation. And there's a variety of physical processes that can cause fractionation among isotopes. So things like evaporation, diffusion, freezing, changes of state. Lighter molecules, smaller, lighter molecules tend to evaporate faster. And so if you're the light isotope, you might evaporate faster. Heavy isotopes tend to condense out faster, so they rain out of the sky, things like that. At colder temperatures, isotopes might be more heavily fractionated, but there's also a variety of chemical and biological processes that can cause fractionation, and these are really interesting as ecologists. So some reactions and enzymes will discriminate against the heavy isotope, and then we can follow that, we can track that through a bunch of tissues. Some are slightly slower in chemical reactions, and some isotopes bioaccumulate in tissues. So we're going to talk about all of these things. So first of all, we'll talk about the water cycle, because there are both hydrogens and oxygens in water. And there are different fractionations happening at all of these different steps of the water cycle. And we can use water isotopes to help track things like the direction of the prevailing, weather. This map shows you the oxygen 18 isotope concentration in a delta O18 per mil value, ranging from greater than 2 all the way down to -18. So there's a lot of negative numbers when we're talking about isotopes. What was really cool about this is that surface water, if you just took some surface water from a stream and you ran it through an isotope ratio mass spectrometer, we could predict where it came from based on this map, right? Warmer regions tend to be less depleted, less negative. So the bluer, kind of warmer, greener colors, those are less negative than the colder northern parts of the country. And these isotopes persist on the landscape. So really useful for things like migrating birds. So the isotopic signatures of birds, bird feathers, bird blood can tell us where they've been living, where they've been coming from, where they've been getting their water. And so some really cool studies using oxygen isotopes and bird migrations. Here's some more information. Both hydrogen and oxygen isotopes are really valuable when it comes to studying water. And so we can tell where a plant is getting its water or where a stream's water is coming from. So different types of water will have different isotope signatures. So the groundwater versus surface water versus rainwater versus snow versus glaciers. And so if you take a water sample, you can run it off to the lab and ask them what the isotope concentration is, and that might give you an idea of where the water is coming from, what kind of pool the water was most recently in on land. So for example, say you have some trees and you take some leaf tissue and you're wondering where they were getting their water. If they're getting their water from, say, surface water, or if they're being irrigated from groundwater, you might be able to tell the difference between those two things. So these little green pieces of text are for you to use. Here, I'll show you. You've got a little worksheet, and you have nitrogen and carbon, you have either oxygen or hydrogen. And so these little examples are you know, to cut yourself out these little playing cards and place them on the graph where you would find them based on their concentration. So you'd have a tree, and what would it be kind of if it was getting water from the stream versus what would it be getting if it's getting irrigated water. And I'll show you the answers at the end. So you can try to play along if you want by cutting out those little playing cards, putting them on the graph. Okay, so a little bit more about isotopes in water. You can see O18 values in the ocean, it's 0, but as it evaporates, it becomes more negative. And that's because the heavy isotopes don't like to evaporate. So it's more negative in vapor, but then the heavy isotopes that end up in the vapor do fall out earlier. And so those values in the rain are less negative, right? And the water vapor actually gets more and more negative as it moves over the land surface. And so this is partly why we see those patterns in oxygen across the map. The same is true for hydrogen isotopes. We can see some really big differences between the ocean water, where we see ice caps, and atmospheric water, rivers and lakes and stream channels and things like that. So for example, you could compare ocean deuterium to a cloud deuterium and put that on your little chart. You could compare ocean to a cloud to the rain, and where would you see those values come up in terms of O18. Okay, so some forensic applications. Our drinking water is isotopically different depending on where we're living. And so we can actually, you can test your hair and you can tell where you were drinking water back through time. So if you moved from one part of the country to another part, the isotopes in your hair would take on those signatures as you moved. And so you can actually use this in forensics to tell where a suspect has been. So if someone has an alibi and they're like, I've never been to Arkansas, you could actually tell based on testing the isotope concentrations of their hair, right? Well, actually it shows that you were in Arkansas at this point in your life, right? Something like that. So there's really cool forensic applications to this too. And you can measure it in your hair. Okay, so moving on to carbon isotopes. There are 3 isotopes of carbon that are common. Carbon 12, which is normal, light, carbon 13, which is heavy, stable, and carbon 14, which is even heavier and unstable, radioactive. If you've heard of carbon 14 dating, carbon 14 can be used to date paleo items like bones and wood and things like that.